Hi, my name is Yang. I will be presenting the paper, Diameter, Matching Peptides to Data-Independent Acquisition Mass Spectrometry Data. As we all know, that proteins are indispensable in all biological processes. To understand the roles which the proteins play, one fundamental question is to first accurately identify these proteins from complex mixture. Currently, the most popular method for the protein identification is based on liquid chromatography mass spectrometry, where proteins are digested into short peptides by the enzymes. Here is an example of the produced tandem mass spectrum, where each peak here indicates a particular molecular ion. Now, we briefly describe how a mass spectrum is obtained. Briefly speaking, peptides are ionized and enter the mass spectrometer. A spectrum of the peptide eluting at a certain time point will be taken, which is called an MS1 or precursor spectrum. In such spectrum, each observed peak generally corresponds to one peptide. To further scrutinize what are these peptides, we need to isolate peptides and identify them by further fragmentation. Here, the fragmentation means to break down the peptide into a set of prefix or suffix ion in another spectrum, which is called an MS2 or fragment spectrum. Due to the limitation of instrument, we cannot fragment every possible peaks what we observe. Depending on how to prioritize the peptides to fragment, it can be roughly divided into two categories. The first and most common one is called data-dependent acquisition, where we select a fixed number of peaks to generate MS2 spectrum according to their observed intensity. Although this strategy is very straightforward, the limited and selective sampling may miss potentially interesting information. Another increasingly popular category, which is the focus of the paper, is called data-independent acquisition, where we don't select peptides, but instead bin the peptides into several isolation windows along the MZ axis, and we then generate MS2 spectrum for each window. In such case, the fragment ions are not from a single peptide, but from a mixture of peptides. Such systematic sampling of entire isolation window will not lose any information. However, the resultant spectrum is highly multiplexed and therefore very hard to analyze. The obtained MS2 spectra, regardless from DDA or DIA, are stored for matching against a protein sequence database. We first theoretically fragment each peptide in the database. By defining some similarity measurement between an observed and theoretical spectrum, we can rank the peptides with respect to that similarity and keep the top K peptide spectrum matches. It is worth mentioning that the standard database search methods are generally designed for DDA data. So we typically keep the top one match because we assume each spectrum is generated from one peptide only. There exist a few methods to search DI data. However, existing methods all suffer the following limitations. For example, some methods they perform poorly when the isolation window is wide, where wider isolation window means more multiplex data. And some methods is built on deep learning-based spectrum predictor, which is trained with instrument-dependent data and with limited support to post-translational modifications. And some methods extract DDA-like pseudospectra from DIA data, which will fail when there is no detectable signals in the MS1 run. Therefore, we propose Diameter to tackle all these limitations. Here is the workflow of Diameter. Briefly speaking, Diameter is based upon a standard DDA search engine, but with an extra PSM filtering step. Next, I will explain each step in more details. The input to diameter is a DIA data and a peptide database, just like DDA search, without depending on any spectrum library. Diameter will search the DIA data with a standard DDA-oriented search engine, TIDE. TIDE measures the similarity between an observed and theoretical spectrum using X-core score, which is a variant of the inner product. For each spectrum and each charge state, ranging from 1 to 5, we will save the top k PSMs. In this study, we just chose k equal to 5. Recall that in DDA setting, we only keep the top 1 match, but now in DRA setting, we keep top 5 matches here, so we expect that many of them will be wrong. Therefore, we introduce a key post-processing step to eliminate potentially wrong PSMs. In particular, we define a composite score for each PSM, which is calculated as the weighted sum of 5 features. I will elaborate these features later. We re-rank the PSMs by this new score, and we enumerate the re-ranked PSM list from top to bottom. We will retain every PSM we encounter until we hit the one corresponding to the largest X-core score. In other words, we will treat the composite score of the PSM with largest X-core as the baseline, and we will discard those PSMs whose composite score cannot beat this baseline. Now we visit each features used in the composite score. As we are all familiar with, X-core score measures the fragment level matching evidence. Here, the more peaks are matched between the observed and theoretical spectrum, the better it will be. The precursor intensity measures the precursor level matching evidence. That is, if a peptide is matched based upon the MS2 spectrum, we hope to also find its supporting evidence in MS1 spectrum accordingly. The fragment matching p-value measures how likely the theoretical fragments are matched by random chance. For example, in the extreme case shown on the right, because the observed peaks are so dense that the matched theoretical peaks are not as reliable as the left. The rotation time score measures the temporal matching evidence. By using existing machine learning-based method, we can reliably predict the rotation time of peptide sequence. We hope that the predicted rotation time can align well with the observed rotation time encoded by the matched spectrum. And lastly, precursor and fragment co-elution measures the chromatogram level matching evidence. If peptide is real, the chromatogram of its precursor and fragment ions should exhibit a bell-shaped like elution pattern, and we use their correlation to quantify its co-elution evidence. After that, we use Percolator, a semi-supervised algorithm, to re-rank this PSM by their corresponding Q value. To be specific, Percolate works by iteratively training a series of support vector machine classifier, where negative examples are decoy PSMs, and positive examples are high-scoring target PSMs. We noticed that Percolator is designed for DDA data, so we made a couple changes to customize Percolator for DI setting, which is we assign all PSMs related to the same target decoy pair to the same cross-validation split. We also select the top-scoring PSM per target decoy pair. and we calculate its own peptide level FDR estimates. We also notice that it is unfair to directly compare different peptide detection methods because each method chose to estimate false discovery rate in its own fashion. Therefore, we use an entrapment strategy to make consistent FDR estimation across methods by using the same set of decoys. We notice that these pseudo-targets or decoys lead to loss of statistical power, so we only use that for evaluation purpose. We systematically evaluate diameter on different datasets with diverse settings, from different instrument types, narrow or wide isolation window size, fixed or variable isolation window paradigm, etc. For example, in this yeast dataset, which we generate with enriched methionine, we vary the isolation window size from 5 mZ, 10 mZ, up to 20 mZ. For another example, in this well-studied LFQ bench, we not only use two different Chiptov instruments, each of which we also use either fixed 25 mZ or variable isolation window paradigm. We compare the performance of diameter against other three existing methods in terms of the detected peptides at 1% FDR. The FDR is estimated by the entrapment strategy mentioned earlier. Not surprisingly, we found that the Chiptov 6600 provides improved peptide detection over its predecessor, the 5600. Also not surprisingly, we found that employing a wider isolation window drastically increased the complexity of the resulting spectra, and thus in turn decreased detection sensitivity. Overall, we observe that diameter confidently detects more peptides in most cases. In DRA analysis, the huge dynamic range of protein abundance in complex samples means that some peptides without detectable MS1 signals may still be detectable based on their MS2 signals. Such peptides may be problematic for methods such as DI Empire, which is designed to construct pseudospectra based on MS1 signals. Here, we showcase some confidently identified peptides detected in different instruments. These peptides report low Q values, and diameter can confidently detect them with both +1 and +2 isotope signals, but without any monoisotope signals at all. We next evaluate the marginal importance of each diameter feature. We ran a control study in which we modify diameter to include various subsets of the full collection of features. In particular, we consider 10 variants of diameter, 5 in which we eliminate one single feature, and 5 in which we retain only a single feature. In each case, we run diameter using the reduced feature for edge rescoring, edge filtering, and Percolator post-processing. The results of such analysis suggest that all 5 features contribute positively to the diameter performance. We also found that the utility of some features appear to vary by instrument. For example, the retention time score make a negligible contribution to Orbitrap instrument, but notable contribution to Chiptov instruments. Lastly, we demonstrate that diameter accurately controlled FDR by comparing the nominal Q value reported by each method against the Q value estimated by the entrapment strategy. The results suggest that 3 of the DIA methods, including diameter, tend to be conservative, in the sense that the nominal Q values are larger than the pseudo-target Q values. The results also suggest that these 3 methods, including diameter, could potentially gain more statistical power simply by improving their method for FDR estimation. And finally, to conclude, diameter is more sensitive than other existing methods without relying on spectral library. Diameter is robust in different settings, and diameter can detect peptides even without any detectable MS1 signals. Finally, we want to emphasize that both the source code and the software can be available as a part of Crux toolkit. Thank you.